Okay, continuing on with Mendel and genetics. Mendel was famous for coming up with three laws. One of these laws we've already discussed. We talked about how one allele, whenever it is present, so long as it is present, it is always going to show itself. We call this the law of dominance. If you recall, we said that tall was dominant to short in pea plants. So anytime a pea plant has the dominant allele, which is labeled by big T, that plant is going to be tall. Homozygous tall and heterozygous tall. Anytime the recessive allele, or the little t, is present, if the big T, or the dominant allele, is present, it will cover the recessive allele. So the only way to have a short plant, again, is to have two recessive alleles. Now, Mendels Law of segregation, what it says is a little bit different than law of dominance, obviously. And what it says is when you're forming gametes. Remember, in humans, gametes are eggs and sperm. In a plant, such as pea plants, we call it an egg and we use pollen instead of sperm. Now, what this law says is, the law of segregation, it says that when you're making gametes, it says that alleles are going to segregate from one another and that you will only put one allele into the sex cell. Now, if I show you some chromosomes, if I draw some here, we'll pretend like this is for a pea plant. There's the number 1 homologous chromosomes from each parent. There's the number 2 homologous chromosomes. I'm going to put some genes on here, or alleles. I'm going to say that that's big T, this is little t, and we'll say that this is big P for purple, and this is little p. What the law of segregation says is that when making a gamete, and here's my gamete, that the alleles are going to segregate from one another, meaning big T and little t are no longer going to be together. I'm going to segregate them and only put one of these chromosomes and one of these alleles, because there's the one allele per chromosome here, and I'm going to randomly put the big T into this sperm. It has segregated itself from the little t. You no longer have the diploid chromosome number. You now have a single chromosome here, and I segregated the two alleles from each other. If you recall where this really takes place from when we did meiosis, in anaphase 2, we've got chromosomes. This would just be a picture of anaphase 2 here. The one with the big T is heading to one side, which will become a sperm, and the other with the little t on it is heading to the other side of the cell. And when this pinches off, you end up with two sperm cells. So the law of segregation can basically be explained through anaphase 2 of meiosis. Sometimes those stick together and create a sex cell where you have double the number of alleles you need, and that can cause some real problems, and we'll talk about that later. Mendel's law of independent assortment is a little bit different. And what I'm going to do to explain that is I'm going to again draw some chromosomes. This is the number 1 pair of homologous chromosomes. We'll say that this is big T and that this is little t. We'll say that really close to that on this chromosome is going to be the allele for flower color. So we'll say that this is big P and this is little p. And I'm totally making up randomly. There's no reason why these are both dominant and these are both recessive and on different chromosomes. The little t could be over here, the big P could be over here. I just totally made this up. Over here on this chromosome, I'm going to put little w here, and at that exact same spot, big W is going to be here. So what Mendels Law of segregation says, and again, this is about when forming gametes or sex cells, we already know that because of the law of segregation, that I can only put one of these two chromosomes for each one of these into this sperm. I'm going to go ahead and I'm going to pick this one randomly to put into this sperm. So here's that chromosome. There's the little t. There's the little p. I randomly am going to pick one of these two chromosomes. I'm going to pick the one that's got the big W on it. And I'm going to put it into here. I just did Mendel's law of independent assortment. And what that law says is that when making gametes, alleles that are on different chromosomes will assort themselves independently from each other. Now, where you have to be careful with this is the key to this is it has to be alleles that are on different chromosomes. Because big T and big P, I'm sorry, I use little t and little p. Little t and little p they are not going to segregate from each other, or they are not going to assort themselves independently from each other. These two are going to travel together because they are on the same chromosome, and because they're on the same chromosome, they're going to end up in the same sex cell. Where the law of independent assortment comes in is that when they talk about alleles on different chromosomes, just because this chromosome went, it has no bearing whatsoever on which of these two are going to get sent into this sex cell. These two alleles, they will get sent together because they are right next to each other on the same chromosome. But again, these two alleles have no bearing on which one of these are going to get put into that sperm. And this is another guarantee of diversity because the randomness of which chromosomes get into what sex cells is so random.